The expansion and contraction of alveolar sacs is a high-speed mechanical process driven by a deeply vascularized system of deliberate force. The lungs possess no internal skeletal musculature. No part of the lung tissue is capable of directly pumping or drawing in atmospheric air. Instead, the lungs sit within the thoracic cavity, a sealed pressure chamber where ventilation follows the laws of gas mechanics. Breathing is a passive physiological consequence of creating specific internal pressure differentials. The respiratory system serves as the body's fastest chemical buffer, using gas exchange to regulate the bicarbonate buffer system. The rate of carbon dioxide expiration acts as a master dial for blood acidity, providing a constant defense against systemic acidemia. We'll map the biophysics of ventilation, derive the biochemistry of pH control, and analyze primary experimental data to see how pathology destroys equilibrium. Survival depends on the link between physical tissue compliance and chemical homeostasis. Two membranes define the boundaries of the thoracic cavity, the visceral para lining the lung and the parietal pleura lining the chest wall. If you push your fist into a partially deflated balloon, your fist represents the lung, while the balloon layers represent the pleural membranes. These layers create the intrapleural space. It is a potential space filled with lubricating fluid that locks the lung to the chest wall. Boyle's law dictates that as a gas's volume increases, its pressure decreases. As the diaphragm flattens and the chest expands, intrapleural volume increases, causing pressure to drop. This vacuum drags the lung tissue outward, sucking atmospheric air inward to equalize the system. Inhalation is an active process requiring ATP to contract the diaphragm, but exhalation is passive. Once the muscles relax, the natural elastic recoil of the lung tissue snaps the chest back to its resting state. Human ventilation is an exercise in negative pressure mechanics, governed by the preservation of elastic tissue. Quantifying these mechanical pressure differentials is a clinical necessity for detecting respiratory failure. A spirometer translates air displacement into a graph of specific lung volumes. This oscillating wave is the tidal volume, the amount of air moved in a normal resting breath. Forced inhalation and exhalation define the expiratory reserve volume and the inspiratory reserve volume. Together, these metrics form the vital capacity, the total air volume the lungs are capable of moving. The space at the bottom of the axis is the residual volume. This is the air that prevents alveolar collapse. It can never be exhaled or directly measured by the spirometer. Total lung capacity is a calculated metric that relies on this hidden, unexhalable residual volume. Physical air displacement drives chemical equilibrium within the pulmonary capillaries. This is the bicarbonate buffer system. In metabolic acidemia, hydrogen ion concentrations spike aggressively. Le Chatelier's principle demands the system counteract this spike. The equilibrium shifts left, generating excess carbon dioxide. Acid-sensing chemoreceptors in the medulla oblongata detect this change in blood chemistry. The medulla signals the diaphragm to hyperventilate. This blows off the excess carbon dioxide, pulling hydrogen ions out of the blood and raising the pH. The respiratory system is the body's frontline defense against acidemia. Physical damage to the lung tissue immediately threatens chemical survival. Inhaled particulate matter, such as aerosolized crystalline silica, can bypass mucosal defenses and infiltrate the deep lung. Chronic silica inhalation induces oxidant-antioxidant imbalances, leading to pulmonary fibrosis. Researchers analyzed the antioxidant enzyme SOD3 to determine its role in protecting the pulmonary vasculature against oxidative stress. The experiment compared the lungs of wild-type mice against mice genetically engineered to lack SOD3, with both groups receiving a tracheal silica injection. This is a high-stakes stress test. By removing the SOD3 defense, we can observe how silica destroys the biophysical equilibrium of the lung. This chart tracks right ventricular systolic pressure, an indicator of pulmonary hypertension and cardiovascular strain. There is a severe spike in pressure within the SOD3 knockout group. Without the enzyme, silica triggers immediate pulmonary hypertension. This chart tracks SOD3 mRNA expression to establish the molecular response to the toxin. Silica exposure suppresses the protective SOD3 transcript, even in normal wild-type mice. These results are explained by Figure 3, which quantifies collagen levels in the lung tissue. The massive spike in collagen production in the knockout group matches the spike in right ventricular systolic pressure. Without SOD3 to neutralize oxidative stress, silica triggers a massive overproduction of collagen, causing high blood pressure in the lungs. Microscopic tissue analysis reveals the physical location of this collagen accumulation. This manifests as pulmonary fibrosis, thickening the delicate one-cell thick walls of the alveoli. This rigid tissue growth neutralizes the structural compliance required for passive exhalation. Because the lung can no longer easily change volume, the pressure differentials required by Boyle's law fail. The right ventricle is forced to pump harder to overcome this resistance. Simultaneously, thickened alveolar walls block the diffusion of carbon dioxide. This paralyzes the bicarbonate buffer system and risks fatal acidemia. The physics of ventilation and the chemistry of blood buffering are dependent on microscopic tissue compliance, an equilibrium that is both life-sustaining